So I work on a variety of different biological problems, and they all have one thing in common. They have to do with the problem of protein folding. So what's the problem of protein folding? Well, proteins do just about everything in our cells and for us in our bodies. When you think about seeing an apple on a tree, the light that you perceive that apple by is being harnessed by protein receptors in our eyes. And it's a series of actions of various proteins that transmit that signal from our eye into other parts of our brain that can wind up interpreting that image. When we go to pick that apple, our arm is powered by muscle. Muscle is protein. The engine for creating the energy, the mitochondria inside of our cells, are made up of thousands of different proteins that are involved in providing that energy from that movement. When we bite the apple and we taste it, the taste is being perceived by receptors on our tongue and in our nose that are made up of proteins. And when we digest the apple, the enzymes that are involved in digesting it are proteins. So proteins are us, I guess, is the way to put it. And proteins, however, are encoded by DNA. DNA is the code of life, the double helix. And DNA is linear. It's a long, very complicated code, but it's sort of like not really, in some ways, all that interesting in and of itself. It is the information for life, but rather like a cassette tape. It can play different tunes depending upon what part of the DNA is being accessed, but the tape itself is linear. Now, the way this DNA plays different tunes, basically, is that it's decoded into long linear strings of amino acids based upon the sequence of nucleotides in the DNA. So here are two very different proteins that are being made by this different parts of the DNA. And in order for those proteins to function, they have to fold up into these incredibly intricate, complicated shapes. This is hemoglobin here, the protein that's involved in carrying oxygen around in our blood. And this protein over here is green fluorescent protein. It's a wonderfully useful tool in biology. It actually comes from a jellyfish, but it has this lovely property of glowing green when you shine blue light on it. And we use it as a tool, as you'll see a little bit later in the talk. But you can imagine that it's kind of complicated for these proteins to fold up into these shapes. But the problem becomes much, much greater inside the crazy environment of a living cell, where the concentration of other proteins is about 300 milligrams per mil. Now that's about the concentration of protein that crystallographers have in packed crystals when they are actually exploring the protein structure. But of course, it's not like a packed crystal. The cell is very, very dynamic, and proteins are moving around and colliding into each other all the time. So when you think about proteins going about their business inside of a living cell, it's not like Fred Astaire and Ginger Rogers. It's more like the characters in a Marx Brothers movie, where chaos is poised on the precipice of disaster.
Come on.
So that's what we folks in the protein folding field call going off pathway. And proteins are doing that all the time in living cells. They're trying to fold, and they often get it right, they often don't. And disaster can ensue when they don't get it right. And in fact, it's the cause of a lot of human diseases. I'm going to be telling you about a very interesting disease caused by protein misfolding in just a minute. But first I want to tell you where and why we study this human disease in a very unusual, unexpected place. So this problem is as ancient as life itself, and the solutions to it were actually arrived at very early on in evolution. And they're shared amongst all organisms. They involve things like protein chaperones, osmolytes, protein remodeling factors, protein degradation machinery, and compartmentalizations of proteins. And it's a pretty complicated problem, so our motto is, if you've got a really complicated problem that can give rise to a lot of very difficult diseases in human beings, for example, be a good place to start someplace simple, good thing to start someplace simple, and then move around a lot. So we very often start our experiments with this organism here. This is the budding yeast, known as Saccharomyces cerevisiae. And it's actually been an incredible friend to mankind. Man has been taking advantage of this organism for several thousand years because it likes to take sugars and turn it into alcohol and CO2. And that means that we have used this organism to make bread, and we've used this organism to make beer and wine for thousands of years. Now, about 100 years ago or so, brewers were facing the problem that sometimes they'd brew a batch of beer and it would be great, another time they'd brew a batch of beer and it would be terrible. And so they decided to start working on the biology of what was going on in these brewing vats of beer. And they found out that there was this particular organism that was responsible for changing the grain into alcohol and CO2. And they then started, more and more work was done on this organism, originally with this goal of brewing better beer. But it turns out that as biologists studied this organism more and more, we found out that it has a tremendous number of properties that make it just ideally suited for biological research. For example, it can grow either in the haploid or the diploid state. And we are obligate diploids. We have one set of chromosomes from our mother and one set of chromosomes from our father. Until that sperm and egg comes together, the sperm itself is the haploid, the egg is the haploid. They're not doing anything much. It's only when they come together that you form a new organism. But in yeast, they can live, and you wouldn't really know much difference between them being the diploid state and the haploid state. But for a geneticist, that's a dream. Because if you have one bad copy of a gene and one good copy of a gene, you can keep that bad copy in a diploid state so that the good copy takes care of the cell. Then when you sporulate the cells and you get the haploid progeny out, you can see what that mutation does. It's really a wonderful property. Yeast cells also have very small genome, so it turns out that this was the first higher organism to have its genome sequenced. Now you may be wondering why I would call this a higher organism. It doesn't seem like a higher organism, but it actually is. It has much more in common with human cells than bacterial cells do. And the reason is that these yeast cells have a membrane-bounded nucleus. They contain mitochondria, which are energy factories for the cell. They contain a whole series of membrane-bounded compartments where they put different proteins are compartmentalized and secreted from the cell or not secreted from the cell, moved around the cell in different ways. And that's a lot like our cells work. So even though these cells are separated from us by about 1 billion years of evolution, they actually do things in a remarkable number of details. They do them very much like we do. So we often use these cells of living test tubes to study really complicated problems in protein folding biology. And that takes us into problems of evolution. It takes us into the problem of human diseases. And so I'm going to actually bring two of those problems together today by telling you about some work we initially started to do because we were interested in a very bizarre human disease, that then when we got into yeast and started studying this process, took us into a very interesting realm of evolutionary biology. So the disease that I'm going to be talking to you about is a neurodegenerative disease. It's a really dreadful one. And it's also a rather mysterious one. It's one that we still don't understand completely, although we're making a lot of progress on it. This is a front page from my hometown newspaper, and as you can see, imported cows killed, brain ailment a mystery. This was from back in the days when Great Britain was staggering under the mad cow epidemic. It was very, very frightening because we didn't know. It became apparent that the disease was actually crossing the species barrier, and some young people in Great Britain were coming down with the disease. But thank goodness there's a very strong species barrier, and so really only a small number of people have come down with the disease so far, and it looks like it's not going to be of outrageous proportions. This is a very, very bizarre disease in many different ways, and I'm just going to tell you a couple of them. So most diseases, most infectious diseases that we get are caused either by viruses or by bacteria. So these viruses and bacteria have their own nucleic acids, their coding information of life, that they carry with them, and they use that to subvert to take advantage of our bodies and to make more of themselves. This particular disease, the diseases known as the prion diseases, can be manifested as infectious forms, but you can also get the same disease by inheriting a mutation in a particular protein in your body. Moreover, you can also get this disease just by sheer spontaneous chance, and that usually happens in the elderly, and thank goodness it doesn't happen very often. But there really isn't any other disease that I know of that has these properties. It's really quite strange. And the really strange part about it is that the spontaneous forms of the disease and the heritable forms of the disease can both give rise to infectious material. Now, when Stan Prusner was studying this disease a couple of decades ago and trying to purify the infectious material, the more and more he purified it, the more it became apparent that it looked like it was just made up of protein without any nucleic acid, without any code in there. So how could this protein possibly cause a disease and moreover replicate inside of our bodies and give rise to more and more infectious material? Well, the solution to this turned out to be solution to this problem that this infectious agent might be a protein is that it's one of our own proteins. So it's a protein that we already have in our brain. We're expressing it all the time. And it has a normal conformation that's obviously diagrammed in a very simplified way here. And thank goodness it normally stays in that conformation. Every once in a while, however, it can get into this altered conformation down here, which is really a profoundly different conformation. It folds in a very different way. And it has a sort of uncanny ability in that changed folded state to be able to entice the other protein to change its shape too. And it's that process that generates a sort of protein conformational chain reaction that generates more and more infectious material, and in the process, in a way that we still don't understand, winds up killing our neurons. Now, we think, based upon some work that we've been doing in yeast cells actually, that it's not that this protein in this shape here interacts with the protein in that shape here, there directly and gets it to change its shape. But rather we think that this protein transiently exists in an unfolded state. Every once in a while it folds and unfolds, and it's when it's in this unfolded state that it's susceptible now to being templated by this altered protein to generate more and more of this altered state.